Welcome to this week's meeting of the Rotary E-Club of Silicon Valley. The theme of our club is innovation, entrepreneurship, and education. And our club specializes in using technology in our club for all of our meetings and our ancillary things. Now, we have a very special guest today, which is one of our members. And she's going to give the first one of, I hope, several presentations on the area of medical research. And Ferheen has come back from Japan recently, where she has finished her master's degree and has conducted her research, of which she is going to tell us about today. And go ahead, Ferheen.
All right, thank you so much, Roger. Hi, everyone who's listening to this recording, and of course, Roger. So I will switch to my PowerPoint. I made a little slide. Oh, hold on. Okay, hold on. So I'm going to share my screen. Okay, hopefully you all can see that. And now I'm going to begin my little PowerPoint presentation. So hi, everyone. My name is Ferheen. And as Roger mentioned, yep, I recently came back from Japan. But let me tell you a little bit about myself and that I will be presenting about the CRISPR-Cas9 system today. So here's a few pictures of myself. So I graduated from UC Berkeley in 2014 with 2 degrees, one in molecular cell biology and another in Japanese. And I had the most wonderful opportunity to do my master's degree in Japan. So I received the Japan governmental scholarship, and I was there for 3 years at Osaka University, and I did my master's in reproductive biology. But first, I want to show you a few pictures of myself because, I just thought that was a lot of fun. So this is myself in the first picture is myself in Japan, and specifically in my lab, working on one of the microscopes. And then the picture next to that is Mount Fuji in the background. Next to that, I got to wear a yukata, which is a very traditional Japanese summertime outfit. So when you go to festivals, you wear it. And then the one underneath is Himeji Castle, and during cherry blossom season, so Japan is just one of the most beautiful countries in the world. I definitely recommend going for cherry blossoms. And then next to that is myself at my graduation day. I got to wear a kimono and hakama outfit, which was, again, very traditional and very awesome. And then right next to that is Osaka Castle, one of my most favorite places. And then finally, the Fushimi Inari Gates, which are again, super, super famous. So if any of you who are listening, are really interested in this scholarship, please let me know. You can put a comment, you can email me, but I also put the link for this scholarship. So, but enough about me. Now I'm going to talk to you a little bit about my lab and then what exactly is this CRISPR-Cas9. So I had the awesome opportunity to be in Professor Ikawa's laboratory at Osaka University. So basically my lab is focused on studying the mechanisms of reproduction and specifically using genetically modified mice. So what that means is that we want to find what genes are essential for fertility in mice, and then hoping that can translate into humans, because reproduction is for the most part a conserved system in mammals. And the way we did this is that we used this CRISPR-Cas9 system to make the mice. And I will I'll explain a little more about what that CRISPR-Cas9 means. And so the goal that my professor has is he wants to knock out all of the genes that are related to male reproduction. And so now let me talk to you a little bit about what does this CRISPR-Cas9 mean? So CRISPR-Cas9 is this gene editing tool that allows scientists to manipulate the DNA of an organism. So this technology was actually, partially founded at UC Berkeley by Professor Jennifer Doudna. So, go Bears. And so, and we use this technology in my lab. And so let me, kind of explain what exactly this means. So there is this Cas9 protein, and I can take it like a pair of scissors. And so what it does is that it can specifically cut DNA at a very specific region, and you can program that region. So as you can see here, there's this guide RNA. And the way that I want to kind of describe it is, when babies have those kind of blocks and cutouts, right? So think about that guide as kind of the block, the shape of the block. And then you kind of have to match it to where, that cutout is and kind of have it fit, right? So the moment it fits the DNA, then this Cas9 protein cuts it, okay? So that's kind of just how this CRISPR-Cas9 system works. So then after those scissors cut the DNA, the cell itself, or the organism itself, has an internal repair mechanism, and it tries to fix the damage, but it can't fix it perfectly. So what it ends up doing is that it has insertion or deletions. So again, there's a mutation that happens there, or there's a specific point mutation that can happen. So there's maybe something is very specifically done. But again, but this system is not perfect, and it leads to mutations, okay? And so it can't repair it 100% properly. But the good thing about that is, so hopefully then the protein that gets created becomes inactive because of that mutation, right? So, let's say, the protein starts forming, but then suddenly because of the mutation, it stops because there's a mutation, it can't continue on. So what that would do is that now the protein can't, function properly, and hopefully this leads to a phenotype or observable characteristics. So now here are some examples of what that means using, again, this CRISPR-Cas system. So the first one is actually a paper that was published in 2016 from my laboratory. And in this specific case, so as you can see, you have the wild type mice, the baby mouse, and this is normal, right? So it has a very normal head shape. And then in this next one, it has one copy of the mutant gene, right? Because we have two copies of the genes. But because of the wild type gene is more powerful, it's stronger than the mutation, it also has, fairly normal head shape. But if you look at the mutant, the complete mutant, it has this kind of weird head shape. And actually, this is hydrocephaly. And what that means is that the brain isn't formed properly, and unfortunately, this mouse dies. So using the CRISPR-Cas9 system, basically, our lab, we mutated this gene, and it caused, unfortunately, the mouse to die. In this case, this is another lab in 2017. They used the CRISPR-Cas9 system to show that, when they mutated a specific gene, it, went from standing really tall to the plant falling down. And then finally, in this case, in 2016, they used CRISPR-Cas9 to change the color of this zebra Daniel fish. So the CRISPR-Cas9, again, used in pure, this kind of pure basic science, you can mutate a gene and then see what happens to it. So to continue on, how can we apply this CRISPR-Cas9 to humans, right, in medical research? So first off, as a plug of my lab. So again, my lab is a reproduction lab. So we used CRISPR-Cas9 to find candidates for male contraception. So very specifically, my advisor in 2015 published a really, really awesome paper where he found that this there's something inside the sperm flagella, right? It's called calcineurin, and it's in the mid piece of the flagella of spermatozoa. And he found that if you knock it out, the spermatozoa can no longer swim properly, which leads to infertility. So what he found actually is that this calcineurin, there are already drugs that target calcineurin. And when the drug is applied to mice, they can no longer have babies. But when you stop applying the drug, they can. So it could be a really cool new form of contraception. In another case, let's see here. So immune therapy. So this is a really interesting one where these scientists, they took cells, living cells from humans, and they edited them using CRISPR-Cas9 and then put them back. So this is a really interesting, way of using CRISPR-Cas9 for, again, immune therapy. And then in one more case, sickle cell. So hopefully, folks who are listening to this program know what sickle cell is, but I'll give a really brief introduction to it. So sickle cell, first off, normal red blood cells have that kind of circular shape, and it can, pass through capillaries very normally. However, sickle cell is a disease that causes mutation of the red blood cell shape, and it becomes kind of that crescent sickle shape. And it leads to blockages. So it's harder for sickle cell cells to go through. And that causes anemia and a whole load of different other problems. However, so these scientists are using the CRISPR-Cas9 system to correct the genetic mutation that is responsible for sickle cell. So they're trying to see if they can just cut out the gene or again mutate the gene again to cause it to undo that sickling shape. So this CRISPR-Cas9 system is a super interesting way, super awesome way to take genes, mutate them, and then again kind of fix things, fix diseases. And I think that one of the reasons why this is so cool is because it is really, really easy to do. It's cool, but also kind of scary that it's easy to do. So, and hopefully, maybe later in the questions we can talk a little bit more about the implications for this. So, but with that said, that again, that's a brief overview of the CRISPR-Cas9 system and how it's currently being used in both basic science as well as kind of medical research to help humans. And so I just wanted to again say thank you and a shout out to my lab at Osaka University. I included the link in this presentation, and most of the information on the website is in Japanese, but obviously all of our articles are in English, so please take a look at our publications. And shameless plug, so I got to write a review article about fertility research using the CRISPR-Cas9 system. So the article is free, so please go ahead and read it if you are interested in what I did and what my lab does and how we use CRISPR-Cas9 in my lab. But with that said, thank you very much, and I open it to questions.
Thank you very much, Ferheen. That was really interesting.
Great.
When you said it was very simple to do, I was really surprised.
Yeah.
Because you're dealing with something that is so small.
Yep.
Could you tell us how you do that?
Yeah. Okay. So if you remember that picture that I have of like me as a really cool scientist, and in the background you see like that egg, right? That's
right.
exactly how we do it. So what we do is that we take the mouse egg, a fertilized mouse egg, we inject it with a CRISPR-Cas9 plasmid, right? Or it's basically just DNA. Think about it that way. We inject it with the CRISPR-Cas9 system, and then it works, and then it does its cutting thing, and that's it. And then we take the egg and we put it back inside of a female mouse, and then the mouse has the baby. That's how simple it is.
Oh, wow. So how would you do it with the sickle cell then?
Yeah. So with the sickle cell, I think that, I think that right now it's still in the trial stages with the mice, right? Because the thing about CRISPR-Cas9 and again, this system is the fact that if you want to correct a genetic mutation using CRISPR-Cas9, you have to do it when the human is just a little cell, right? And it's just got fertilized. And that has a lot of risks, right? But I think in the case of sickle cell, I think that what they did with the mice is, they tried it that way, right? Where they have the mutant gene, and then they using the mouse, babies, right? They made many mutants. But of course, we can't really do that with humans. That's not legal. So, again, I'm not 100% sure how they are going to try to use this technology with humans. But what they could do is try to do it using kind of that immune therapy method of take some of the cells, mutate them, and then put them back, right? So that could be a way that they can correct for it. But yeah, so that's the kind of ethical issues that we're getting kind of involved with using this system, is that you have to start when it's there, just one cell, right? Or like, well, two cells, right? And then go from there, and that can lead to many issues. So, and
Oh,
you
yeah.
can't guarantee it. You can't guarantee that it works. That's another thing. So
Plus, to do it for humans at that time when there's only two cells,
yeah.
I can't see how ever that would be possible.
Yeah, it's yeah. So as of right now, we shouldn't be using it. However, there are groups. There is a group in China that's already started to do CRISPR on human embryos. But in that specific study, they used it on unviable embryos, so embryos that have some sort of other genetic mutation. But they did see that it works on humans too. So it's kind of cool, but also kind of terrifying.
Yes.
Yeah.
First of all, how long ago was this method discovered?
2013. So in 2013, well, I think, so, Professor Doudna, she kind of, and her team, of course, kind of, they discovered the fact that they can use it to do gene editing. The CRISPR system was actually discovered at Osaka University back, I don't even know, many years ago, the fact that this happens in bacteria, right? But it wasn't until Professor Doudna's team and I believe I can't remember her name, but another professor, I believe in Europe, France, I believe. But they kind of worked together on this. But they started realizing that, oh my goodness, we can actually use this as a tool and not just as a system, an observable system that happens in bacteria. So that's, yeah. So it's pretty cool.
Yes.
Yeah.
And has there been any discussions so far about the ethical questions of around using this tool?
So many. It happens all the time, right? So there's many conferences where, these kind of leading scientists kind of sit down and say, how can we make sure that this isn't in the wrong hands? Because again, it's so easy. If you have money really, and you can buy one of those microscopes, it's really easy. So there are many ethical discussions that are going on right now of how can we use this technology more ethically. But unfortunately, nothing has been kind of set in stone, right? There's no 100 rule of we can do this with humans or we can't do this with you. There's nothing yet. So I'm going to be very curious to see what happens in the near future regarding this technology. Yeah.
Right.
Mm-hmm.
It's very, very interesting. I'm very surprised, and every time I come back to looking at what's happening in medicine, I'm amazed at what has happened to date, but I'm also awed by the fact of how much there is left yet to do.
Yeah. It's incredible to know that we still don't 100% understand how life works, and yeah, there's something new to be discovered and every single day.
Yes.
Yeah.
Do you intend in the future to go on and do medical research, or?
I do. I really, so currently right now I want to become a gynecologist, and go back into more clinical medicine. And I would love to do more research with humans and not mice. But yeah, so I am really interested in, again, looking at contraception for humans. But again, on a more clinical level instead of a mouse level.
Right. And what are you planning to become a doctor then, or?
That is what I'm currently planning to do. Yes, I'm on that track right now. I know our group has a few awesome doctors and folks in medical school, so I'm I've been kind of picking their brains a little bit. Yeah, so I am going to go back to Berkeley in the fall, and for the next few semesters to take a few supplemental science classes, and then I'll be taking the medical exam, the test, right, to get your score, and then with that I'll apply to medical school. So hopefully I have a good application.
Right. Thank you very, very much for your presentation. I've really enjoyed it.
Thank you.
And at this point in time, I'd ask our visitors and our club members to fill out the comment section at the bottom and also register your attendance. If you're a visiting Rotarian, then by putting in your correct email address, you will get back a receipt for the attendance at our meeting so that you can give it to your club secretary. Now, as it is our practice, I'm going to ask Ferheen to finish off the presentation with any further comments that she would like to make.
Sure.
Ferheen.
Thank you, Roger. Again, for the folks who are listening, thank you so much. Yeah, I mean, the CRISPR-Cas9 is an awesome, fantastic technology that has just been kind of discovered a few years ago that we can use. Again, it's not 100% ready to use on humans just yet, but maybe one day maybe it'll be common to do this system on humans. I don't know. With that all said, I hope that, this kind of sparks your curiosity for science. And again, if you have any questions, please let me know. And if you're, again, interested in what my lab does or my paper or even just, maybe you want to do your master's or PhD in Japan, please let me know and I will, hook you up with that information. So thank you very much and have an awesome day.
Thank you very much, Ferheen.